Okay, hi everybody. Thank you for coming. This is an invited lecture that's sponsored by the Balance and Falls Research Center. It's actually the first one for our formal new center. And we wanted to thank the School of Kinesiology and Dr. Buchel in particular for the continued support of all of the speaker series that have been going on. I'd like to acknowledge that we're gathered today on the traditional, ancestral, and unceded territory of the Musqueam people. And with that, I'm very pleased to introduce you to Dr. Sommer Amundsen-Huffmaster. She is a assistant professor in the Department of Neurology at the University of Minnesota. She received her Bachelor of Science in Dance and Engineering with a mechanical emphasis from Hope College in Minnesota. Oh, sorry, Michigan. In Michigan, pardon me. And then received a PhD in bioengineering from the University of Kansas, after which she did a postdoc with Dr. Colin McKinnon, who's connected with multiple researchers in our neuromechanical lab, in the movement disorders lab in Minnesota. Sommer's research interests include applying engineering principles to analyze human movement and biomechanics, and looking at how treatments can modify the motor symptoms of Parkinson's disease. And we're very lucky to have her to come here and present on her work of trouble in transitions, the neurophysiology of freezing and gait of Parkinson's disease.
Thanks, Mark, for that introduction. As he said, I'm an assistant professor in the Department of Neurology, but I'm an engineer by training. So I do have at least one obligatory equation, because otherwise they take away my engineering card. So I come to you today from the University of Minnesota. This is where I grew up. I went away for 10 years and came back. And I joined the lab of Colin McKinnon, and I just stayed. He said, the grant's 5 years, and I just bought a house and said, all right, tell me when to leave. And he hasn't told me to leave yet. So I'm still here. And I want you to know that half of the United States mislabels Minnesota as Canada. So we kind of think ourselves as kindred spirits. So the work that I'm going to talk about today has primarily been funded by Colin McKinnon's R01s. I also have some data from an R01 by Noam Harrell and the Udall Center of Excellence. So these are all NIH-funded, federally funded grants in the US. And I have no disclosures. The overarching themes of everything that I want to talk to you about today is really about Parkinson's disease and looking at understanding the coordination and the motor control of complex movements such as gait and balance, and especially freezing of gait in Parkinson's disease. And then the second half of my talk, we're going to look at understanding the effects of neuromodulation on gait initiation. I'm going to talk about transcranial, direct current stimulation, deep brain stimulation, and electrical vestibular stimulation, which you are all very familiar with here, but it's kind of new for us. Okay, so there are expected to be 1.2 million people with Parkinson's disease in North America by 2030. Parkinson's disease is the result of a lack of dopamine production in the brain. It includes a variety of motor symptoms, and each person with Parkinson's gets a different set of motor symptoms. Some people are more tremor dominant, and they have tremors and rigidity or stiffness. Some people get changes in their voice or speech. Some people get gait and balance problems, and that's what we're going to be looking at today. In addition, some people with gait and balance problems will also get freezing of gait. And freezing of gait is the feeling that the feet are glued to the floor when you're trying to walk. So 2/3 of people with Parkinson's may eventually develop freezing. Some people get it kind of early on, even in their 40s. Other people get it at the very end stage of Parkinson's, and then some people never get it at all. Freezing is resistant to levodopa, so medication treatment, and it's also often resistant to deep brain stimulation. And it can be a primary cause of falls in in Parkinson's. So if you imagine that you're trying to walk forward and your feet stop, but your center of mass keeps moving, that's a real problem, and it can be really damaging to people. Freezing can be exacerbated by an increase in cognitive load, such as dual tasking, anxiety. So you froze on those stairs the last time, and now you're going to freeze every time you go down them, or sensory input. And I'm going to try to make a case to you today that it's triggered during self-initiated movement state transitions. So we're going to talk a lot about movement transitions. And for these reasons, it's important that we understand the pathophysiology of freezing of gait and that we can start to develop treatments to alleviate or prevent freezing. Okay, so here is my freezing of gait video. Okay, so when people come into our lab and I ask them whether or not they have freezing of gait, sometimes there's confusion between feeling very slow and actually getting an actual freezing. So here this person's trying to walk through a door, and you can see that their freeze is accompanied by trembling of the knees and small, short shuffling steps. Those are kind of like good markers that that's a true freeze and not just akinesia or lack of movement. This person's trying to turn at this line, and he has a pretty long freeze here. You can see that right leg is trembling as he's trying to get it around, and then he goes back to walking. This third one is also a turning freeze, but it's much shorter. So he's going to turn at the line, and you see that brief kind of multiple right steps. That counts as freezing. And then this one is during a gait initiation task. And this person is trying to start walking with his right leg, and he actually gives up and walks with his left in order to try to initiate that movement. So freezing episodes and falls typically occur during self-initiated movement state transitions. Examples of this are starting to walk, so going from standing to walking, turning, stopping, or going through doorways or trying to get on an elevator. And cues have been shown to improve these transitions, and they work best when they're externally given. And I'm going to show you some data to try to support that. So to study movement transitions, our lab focuses on the transition from standing to walking, which typically utilizes a stereotyped series of weight shifts called an anticipatory postural adjustment, or APA. I'm sure many of you or most of you are familiar with this, being the balance group. But your center of pressure between your feet starts when you're standing. It starts kind of centrally located. And before you can lift that stepping foot off the ground, you first need to push down with it and then shift your weight onto your stance foot so you can lift your stepping foot and propel yourself forward. So here's what an APA looks like in control participants on the left and Parkinson's participants on the right. And so this is during a self-initiated gait initiation task. So there are no cues. We just say, whenever you're ready, go anytime. And the participant is asked to wait 3 to 5 seconds before starting to walk forward quickly on their own. And so here you can see in these controls, you can see that they've got a nice quiet, like steady balancing state. And then they push off with their stepping foot before their stepping foot force goes to 0. And simultaneously, they're unloading their stance leg before that goes to a full loading at that toe off. So, but what we see in Parkinson's is a much smaller and slower APA. And APAs are completely absent about 20% of the time in people with Parkinson's. So this, however, is a Parkinson's effect. So people with freezing have similar APAs to their age and sex matched non-freezing people with Parkinson's. All right, so here is what an example of a self-initiated gait with no APA. And you can see that this gentleman is actually leaning forward before he takes that step. So he's not preparing and pushing off in order to take that step. He's just leaning. And if you look here at slow motion, he's actually going to reach this second white line. So he is leaned pretty far forward, and then he just hopes that he gets a step. This is slow motion, so it's coming. And that's where the step happens. So he's leaning pretty far forward. If you have freezing, you can see that this is a pretty dangerous strategy. If you're just going to lean forward and hope your foot comes off the ground, but it's not reliably doing that, that's a problem. And so here's a gentleman who uses a similar strategy, and he does have a freeze. So you can see that he is just frantically trying to get a foot off the ground in order to make a step so that he can like stop that fall that he's started. So here it is in slow motion. So you can see that he starts with his left, and he actually tries multiple times before just starting to kind of like oscillate between legs and see whichever leg he can get off the floor is the one that he's just happy to not have fallen. So we know that APAs are smaller and slower in Parkinson's disease, but we've also shown that everyone improves with a cue. And so here we have a representative control subject, self-initiated and cued, and a representative person with Parkinson's disease, self-initiated and cued. And so you can see that the control has an APA here. They have that loading and unloading, and it gets larger with a cue. And our person with Parkinson's has a tiny little APA there. It's very small, it's very slow, and it also gets larger with a cue. So everyone improves with a cue. Oh, I had boxes, sorry. And this is a Parkinson's effect, or even just an everybody effect. Like if I gave you a cue, you would have a larger APA. So this led our group to ask, what's the best method to cue? And this work was led by Chahao Liu, a postdoc in Colin McKinnon's lab. This was like right when I started as a baby postdoc in the lab. We were doing this study. And so we had 3 types of cues: visual, acoustic, and vibrotactile. The vibrotactile was placed on the outside of the ankle, on the lateral malleolus. And then we had 3 cue timings. So we had a fixed delay where they heard a boop, and then 3 seconds later, a beep, and they had to walk forward quickly on the beep. A random delay where there was 4 to 12 seconds between the warning and the go. And then a countdown, so boop, 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 beep, with like 1 second between. And then a self-initiated, which I described earlier as we just said anytime. We asked them to wait about 3 to 5 seconds and then walk forward quickly. And you do have to coach them pretty extensively during the practice trials because you'll say anytime and they'll just go. But after a few coaching, you kind of get used to like about how long they'll wait. So basically we found that any method of cueing, visual, acoustic, or vibrotactile, had a greater APA, and this table shows the percentage of trials without an APA. So about 17% of self-initiated trials had an APA, but a maximum of like 2-ish in the cued trials. And the vibrotactile was a little bit worse than the other 2 types, primarily because not everybody could feel it or feel it reliably. So even though they did not have a known neuropathy, it was still a challenging task for them. So, but if you want to cue people, visual or acoustic cues are both really reliable. Okay, so then kind of out of that, a similar study, Dr. Leslie Perg worked in our lab to look at the effects of cueing on muscle activity. And we found that for both Parkinson's and controls, cueing facilitates anterior muscle activity. So here's, this is the step leg and this is the stance leg. And we broke the APA into 5 phases, kind of this baseline, then 2 phases of actual APA execution phase during that unloading of the stepping foot, and then post-execution is like after the toe off. And so early in the APA, anterior muscle activity is facilitated with a cue. And then on the stance leg, the facilitation is throughout the APA on that anterior activity. And then cues also facilitate posterior muscle activity throughout the APA on the stepping leg. But interestingly, they suppress activity on the stance leg later in the 2 phases surrounding toe off. So basically, a cue increases early at muscle activity so that you don't have to compensate with late push-off muscle activity. You've already set yourself up for a successful step if you have a good APA with a cue. So it's basically just kind of describing how we're achieving a better APA. And this is for both people with and without freezing. We looked for differences and we didn't really see any differences. So cues are working similarly. So then we asked, can you self-trigger gait initiation? And this was led by a postdoc, Matt Petrucci in the lab. And so here, there was a warning cue and a go cue. And the go cue was either externally triggered, so after 3 seconds, the go cue just went, or there was a self trigger where the participant would push the button to simultaneously get a go cue, and they were supposed to take a step in response to that go cue. And so here we have a baseline passive, and this is all the externally triggered. So an acoustic trigger with a mechanical assist. They were wearing a little foot orthosis. He was a mechanical engineer. And then the foot orthosis with the acoustic cue. And so all of these had an additive effect as long as that all of the devices were externally cued. But when the person pushed the button to get the go cue, it really didn't change. It looks like it maybe even got a little bit worse. So that cognitive task of pushing the button meant that they weren't able to get an effective APA. So people with Parkinson's, and this is across the board, this is all people with Parkinson's, they have a problem with self-initiated movements, and they also have a problem being able to self-cue themselves. So then the question was, can we overcome the self-initiation problem by allowing them to provide themselves a self-warning and then getting an externally driven cue or an externally driven go cue? So here, the 2 conditions were external warning, external cue. So the system was set up to trigger the go cue at a fixed interval, or a self-warning external cue. So they push their button, they immediately get the warning cue, and then 3 seconds later, they get the go cue. So can they self-warn themselves? And here is the external warning, external cue. And here is the self-warning, external cue. So yes, people can self-warn. And we think that this allows participants to properly prepare for the APA. So they do their cognitive button press task, then they get 3 seconds to prepare and successfully initiate the gait initiation task. And we think that this is through an externally cued neural pathway, which is more preserved in Parkinson's than the self-initiated pathway. And this is, again, not a freezing effect, everybody in Parkinson's. Okay, so this led us to look at how the APA was affecting the size and the timing of the first 2 steps. So we know that there's no problem, there's no group differences in the APAs, but we know that freezing is still a problem during gait initiation. So can we start to identify what's going on? So while there were no group differences in APAs, people with freezing took shorter and wider steps, shown here in this figure A. And then in figure B and C, we show that the impact of a poor APA is felt more in people with freezing. So if you have freezing of gait and you have a bad APA, which is caused by your Parkinson's disease, you are less likely to get an effective step. So your APA is really tied, and you can see in these red lines, your APA is really tied to the effectiveness of your steps. But if you don't have freezing, you can have a bad APA and somehow overcome that and still have a decent step. And so it's like more coupled in freezing. And so this suggests an overly large impact of posture and APA sequencing on step execution. And so then here's a freezing effect. Yay, we're getting there. Okay, so we know that freezing episodes are kind of actually hard to capture in the lab, but we have some where we have captured freezing muscle activity on the force plates. And so what we're seeing here is 5 different trials. And so we've got a non-freezing trial with, this is actually a very short step here, but the participant has an APA. Here's the execution phase prior to the toe off. They actually step so short that they step back on the force plate. And you can see that there. Okay, so then these other trials 2 through 5 are all freezing trials. And some of them do not have an APA. You can see here, no APA, and some do have an APA and APA. But what we're seeing is that at the black arrows here show the toe off. And during a freeze trial, a failed toe off seems to be associated with triggering that freeze. So here the toe off fails and they go into some sort of festination where they're just rising up on their toes. Their knees are trembling, but they're not even getting anywhere close to getting a foot off the ground. Here they have a little bit, like they get a little closer until they finally succeed. And again, this is all festination, trembling of the knees. And so we think that this shows that freezing episodes can occur during trials with a missing or a small APA, and that the failed toe off or a very short step is kind of the first thing that happens during a freeze, and it's followed by festination. And so this leads us to think that one primary cause of freezing may be an impaired coordination of the posture to the locomotor systems. So this was really an important thing when I show you kind of our EVS stuff that we're getting to. Okay, so if we look again at the muscle activity during the APA, this is back to Leslie's paper. And I'm showing this here because it shows a really clear switch is needed during the unloading phase. So this is data comparing people with Parkinson's to healthy control. So this is not a freezing effect, but basically you can see that people with Parkinson's have decreased activity in phases 1 through 3, which are here in the orange square. So their muscle activity is decreased. That makes sense because we know their APAs are smaller. And then in order to compensate for that, they have to increase their muscle activity during the execution phase. And so you can see that that's kind of right around that failed toe off. So if they're not doing that successfully for some reason, we could be triggering a freeze. And so this analysis was done freezers and non-freezers, and there didn't seem to be a difference in the muscle activity during successful steps. But I think it's really interesting that right at that execution phase is when that muscle activity switches compared to controls. So I'm gonna take a little bit of an aside and talk about a cadence transitions study that we've been doing. So we're moving out of gait initiation and talking about a different kind of transition. So in this experiment, we had people walking on a 10-meter long figure 8 track, and we had a whole body motion captured with wearable sensors on the feet, the sternum, the low back, and the wrists. And so we did 4 different trial conditions. So a baseline where we just asked them, like, just walk in a figure 8 in your normal pace, just like you're walking down the street. And then we did another condition where they were cued at 100% of that baseline cadence. The third condition, we cued them at 120% of their baseline cadence. So this is a pretty fast walk. It's mostly achievable, but it is challenging, and you do 2 minutes at that trial number 3, and you get pretty pretty worn out. And then the fourth condition, we did 5 trials where we randomly imposed transitions between the natural and the fast cadence at one of the 5 places on the figure 8. So they can't really predict that when it's coming, and we just say, don't try to predict, just when you hear the change in the tones, just walk to the beat. And so this was with the help of an undergrad in our lab, Ryan Bergert. And so we found some really interesting things. So I should say that the reason that I wanted to do this was I thought that people would freeze. Every time I changed the cadence to a fast cadence, I thought that they would shorten their steps and that they would freeze. And it doesn't really do that. Sometimes people freeze, but they're more likely to freeze when they want to freeze. So maybe they freeze during the turns, and then every time they get to that right turn, they're going to freeze on that turn. And some people were like, wow, I've never walked this good with all of these cues. So we know that cues can improve freezing, but I was hoping that challenging cues would cause freezes. It doesn't really work. But we are seeing some interesting stuff. This is really preliminary data. So if you look at an example, a healthy young control. So Ryan basically recruited all his friends. And if we look at the speed versus cadence plots, and then stride length versus cadence. So when we increase cadence in healthy young adults, their speed increases. And that corresponds to basically a consistent step length. So they just keep their step length the same. They go faster, and they're like, I didn't know that we were coming to do a cardio session. And I didn't know you were coming to do a cardio session either. I thought you'd shorten your step length and preserve some energy. But what do I know? But if we do the same thing with a person with freezing, when we increase their cadence, they do shorten their step length, and they don't increase their speed. And we know that those shortened step lengths can cause freezing. So it's part of the sequence effect. Sometimes if people have really bad freezing, they're walking along and they get freezing during steady state gaits. And as they're walking, their steps get progressively shorter until they freeze. And so I was trying to cause that. While it didn't exactly cause a freeze, we're definitely seeing a relationship change. And so if we look at, this is 6 controls, a mix of older and younger right now, 7 people with freezing, and then I've got 1 non-freezer in this study. So really preliminary. But basically, if you look at the slope of cadence versus gait speed, our controls have a pretty strong slope, and our freezers are pretty much at 0. And so we think that people with Parkinson's and freezing may not be able to modify their gait speed in order to adjust for a forced increase in cadence, and instead they take shorter steps. And so, like I said, this could be related to the sequence effect. And while this is a different kind of transition than the gait initiation that we've been talking about, I think it's indicative that this transition is also impaired in people with Parkinson's and freezing. So just kind of an aside, something that I'm really interested in, just trying to make my case for impaired transitions. So this is the freezing effect. Okay, so just to recap the first half here, cues work best in when they're externally given, and cues are important for everyone with Parkinson's, but may be especially critical for people with freezing of gait because they're less likely to overcome a poor APA with a good step. And so here, just for the people in the room who are interested in rehab, a lot of times for freezing of gait, people are offered self-driven cues. So here's a laser light, you push a button, you immediately get a laser light, and now you're supposed to step over that. And while that sometimes works for some people, I think longer term, it would be best if we started using a self-warning paradigm. So we give our participants, a button press. Maybe they hear a little warning tone or see a warning light, and then they get their go cue. And I think that might help longer term. Okay, so the long-term goals of my work are to identify the neural mechanisms of these impaired movement transitions and freezing, and then hopefully identify neuromodulation interventions eventually. So I'm going to talk a little bit about some early work we did in the supplementary motor area. And so we know that the SMA is critical for preparation and initiation of movements, particularly when those movements are self-initiated. And we think that it's important in the coupling of movement and posture during gait initiation. So our hypothesis was that if we upregulated the SMA with transcranial direct current stimulation, that it would improve the APAs and help people with freezing. Okay, so this was our hypothesis. So again, this was with Chahao Liu, who's a postdoc at the same time that I was. We did a double-blinded crossover study. We had 10 people with Parkinson's and freezing who came into the lab off their Parkinson's medications. They did a 3T MRI in the morning, and we used that to target the SMA. And we placed the active electrode over the SMA, and the reference electrode was placed on the forehead for TDCS. And then they did 2 visits. So they had a sham TDCS and an anodal TDCS. And only Chahao knew which one it was. I could not tell. The participant could not tell. And so we basically did 2 baseline trials. So we did a set of cued trials, and then we did a set of self-initiated baseline. We applied TDCS at 1 milliamp for 10 minutes. And then we collected like 5 or 6 trials at each time point every 12 minutes for an hour. And so we measured the APA, and you calculated the center of pressures. And so basically what we had was that cues made the APAs larger and faster, but there was no effect of the TDCS. So this was a bummer for sure. And this was one of my first papers in Columns Lab, and I took it to all of these conferences and just like, it didn't work. But we did get this published, even though our hypothesis was not correct. And so for all of you out there who have null results, take heart. Sometimes you can still get it published. I think there were a couple of key things for us. The study was really well designed. Because we had those cued trials, we were able to show, look, people with Parkinson's, these people specifically, were able to increase their APAs, but TDCS didn't do it. And so this paper is actually cited pretty frequently. Now, there is some evidence coming out that TDCS is more effective when it's combined with motor practicing. So if we ever went back into TDCS world, I would maybe think about doing that. I also wish we had done some sort of control trial of a different type. So TDCS over the SMA should improve reaction time. We didn't measure their reaction time, and I kind of wish we had, just to make sure that we knew the TDCS was working and anything. But there we go. That was my first foray into non-invasive neuromodulation. Oh, I always forget that I have boxes, sorry. Okay, so then we also are doing some work with deep brain stimulation. So DBS can be implanted into either the subthalamic nucleus or the globus pallidus. DBS in Parkinson's is really effective for tremor and rigidity. It improves those symptoms really reliably. But it has variable effect on gait and balance. There's some evidence saying that globus pallidus DBS may increase step length and cadence, while the STN DBS may primarily increase step length. And so we had a hypothesis that a DBS in the globus pallidus could improve gait initiation, while STN DBS remained ineffective. So for this experiment, we collected 4 people with STN DBS, and they were in a slightly different protocol. So they performed both cued and self-initiated trials. And then in the pallidum, we had 8 people, and they only performed self-initiated trials. This is just part of two larger studies, and we had to cut some things in order to make room for other tasks. And so we had a mix of unilateral and bilateral DBS. So some people had one side, some people had two sides. And then we did 6 trials starting right or left. So, okay, so this plot is a little busy, but plot A shows the STN DBS participants, and plot B is the pallidum. And so here we have off their medication before their DBS surgery. Here we have on medication before surgery, off DBS after surgery, and off or on DBS after surgery. So the first off is the medication status, and then the second one is the DBS status. GP DBS participants just came in post-surgery, so they were always off their medications. They were either off DBS or on DBS. So if you look at the self-initiated trials, these are the teal boxes, and there may be like a small effect here of medications in self-initiated, but generally DBS did not affect the self-initiated movements. If we look at cued movements, cues always improve compared to self-initiated, whether you have DBS or you don't. So here's with DBS, and then here's DBS turned on. So there's a little bit, but it's not really significant. But medications have an additive effect with cues. And so that's kind of interesting. So cues are more effective at increasing APA magnitudes compared to DBS. So, yay, cueing. So, so my question is like, what are the neural systems that may be causing freezing in these transitions? So what's causing the trouble in transitions? We've talked about self-initiation versus cued. We've talked about the impairments that occur right at toe off. We haven't really talked about it, but anxiety and dual tasking can also be a component. And so my new grant is looking at 3 systems, the cortical command, the basal ganglia, and then the posture controller. And so as a representative of the posture controller, we're going to look at the vestibular system. You all are like vestibular experts, so go easy on me. But the idea is maybe there's an overactive vestibular system that's responsible for that failed toe off. And so we propose to investigate the relationship between the vestibular system and the shear ground reaction forces under the feet and or the EMG during gait initiation. And we're going to do gait initiation with and without a cue. So this brought me to UBC. Huge thank you to the Bloom and Carpenter labs for hosting me yesterday and today and training me on all of this vestibular stimulation. It's been really great. And so we're planning to use electrical vestibular stimulation to probe the vestibular system. This is a proxy for seeing how the vestibular system is controlling the transition between standing and walking. And so EVS is a bandwidth limited white noise signal, and it evokes a measurable force response during standing and walking. And so we're going to measure the ground reaction forces in the medial lateral directions, and then we're going to measure the muscle activity of the soleus and the gastroc during AVS. So here's my obligatory equation. So we're going to look at the time frequency coherence between EVS and ground reaction force shears, or EVS and EMG. Coherence is a linear relationship between 2 signals in the frequency domain, and it's represented by a ratio from 0 to 1. And so coherence will be calculated across the length of all of the trials for the frequency bins between 0 and 20 hertz. And we're going to significant whole body responses to the EVS will be identified when the coherence exceeds a 99% confidence limit determined by the number of trials and subjects. And so we're going to have people come in over 2 days, and we're going to capture 80 trials of each kind. So 80 self-initiated and 80 cued. trials. And so the preliminary data from this work comes from Amundsen-Huffmaster, who is in the Bloom lab as a postdoc. And when we saw this data, we were just so excited because it fits really well with our transition story. So basically, when people initiate gait, when young adults initiate gait, you see a really strong coherence between the vestibular system and the ground reaction forces. But you see these low spots here at the onset of the APA and then right around toe off. This is true for older adults. You see a quiet period at the onset and toe off. Freezers, this is scaled a little bit odd, but you can still see the quiet periods here. But people with Parkinson's and freezing of gait seem to have a bleed over. So their vestibular system is overactive both at the APA onset and at toe off. And I hope that I've convinced you by now that like those time points are major transitions that are really important in freezing. And so we're hoping, this is just an N of 2, but we're hoping to collect enough data to show that impaired transitions in freezing may be associated with abnormal vestibular sensory coherence. So yes, we were just really excited when we saw this, and I'm excited to like go home and collect our own data and see if it's working. And then I also just wanted to talk a little bit about another area of the grants. We're going to look at the basal ganglia and the cortex, and another potential like place that freezing might be kind of impaired is perhaps an impaired connectivity between the basal ganglia and the cortical systems might be responsible for the failed toe off. So another aim of our grant is to investigate the relationship between the globus pallidus and the motor and frontal cortex during gait initiation, again, with and without a cue. And so we can actually record from deep brain stimulation electrodes now using Medtronic Percept device. You can stream local field potentials basically to a tablet. And we have some experience doing this during treadmill gait using an earlier prototype called the PC+S. And so this is in the lab of Scott Cooper, also with Chahao Liu. And so here we streamed local field potentials from the globus pallidus while a single participant with freezing of gait was walking on a treadmill. And so as he walked, we had to project squares onto the surface of the treadmill, so just little squares of light. And they act as stepping stones. So we said like, okay, step onto the squares. And honestly, he couldn't really do it very well, but without the squares, he couldn't walk on the treadmill at all because the freezing was so bad. Okay, so there's the cue. We give him a cue and he can walk. But if you look here, the constant color cue are the blue lines. And so he's still freezing quite frequently. And so those trials were the constant color, and some other trials had color changing cues. And so while he's walking, the cue would go from a constant color to like a red stripy color. And this was designed on the treadmill for a completely different reason. But we just said like try to step on those squares. And if the squares were changing color, it seemed like he was paying more attention to them. His number of freezing trials decreased pretty drastically. And then also this is the beta power during the swing and stance phase as he's walking. And so when those color changing cues are being used, you can see that the beta power is modulating at a much larger range. And then in the constant color cues, this blue line, beta is not being modulated with swing stance phase. And so we think that the increased cue salience or causing more attention to those cues during gait was associated first with an increase in gait phase modulation of beta power in the local field potentials. Again, this is from the globus pallidus. And then that modulation may have decreased the freezing episodes. So this is just an N of 1, but it just kind of goes to demonstrate that like cues are changing palatal activity. And I'm really interested to see if the relationship between the palatal activity and the cortical activity is also changing. So one problem with cues is that they lose effectiveness over time. But here we can kind of see that some novelty increases that neural activity and the cue effectiveness. So as I've kind of allured to, the idea for my new R01 is to use cueing as an assay to reliably improve gait initiation and then investigate the relationship between the neurophysiological systems during gait initiation with and without a cue. So aim 1 is going to look at the EVS to the ground reaction forces or EMG coherence. Aim 2 is going to look at EEG primarily, so just cortical coherence primarily. Aim 3 will use local field potentials and the coherence between the globus pallidus to the cortex. And then aim 4 is looking at the local field potentials during a freezing of gait course. So the ultimate goal is to identify a biomarker for freezing and eventually identify neuromodulation techniques that could hopefully improve freezing. Okay, so while I have a soapbox, I just want to remind you all, tell your friends and family that participating in research is important. You all are looking for people to be in your studies, and just a reminder that there's a lot of advantages to participating in other people's research. And so this is just something that I think is really important. So thank you. I want to acknowledge 2 mentors at the University of Minnesota, Colin McKinnon and Scott Cooper, the Movement Disorders Lab Group, the Movement Neurophysiology Lab Group, the UDAL group. We have a P3 program for grant writing at the U of M that I participated in, and it was phenomenal in helping me get my grant actually funded, and then some funding sources. So thank you, and I'd be happy to take any questions. Yeah.
Do you know freezing of gait in people with native cerebellum?
That is a great question. Possibly, but we don't really know.
Because I was just thinking,
Yeah.
a lot of the issues seem to be with movement coordination,
Yeah.
maybe even predicting interaction or something.
I'm not confident enough to tell you that people have or have not done that. I haven't seen a lot of it. There's not a good animal model of freezing of gait. So getting at the cerebellum, I think, is a little bit harder, right? But yeah, I definitely have been pondering that. How can we look at that? Yeah, it's a good question. Right. Yeah. I definitely think possibly. I think like the first step is to just like see if we're seeing the same thing that Roman saw. There's, some like other evidence that the vestibular system might be impaired in freezers, but it's not strong evidence yet. The visual system also might be impaired. Like if you track isocods during walking, people with freezing are looking at very different things. But again, that can be like a cognitive component as well. So I think that it's really going to turn out to be a multi-system impairment, but kind of still motor focused. Yeah. So. Yeah. So during quiet standing, you have a lot of like soleus and gastric activity, maybe a little TA as you're standing and swaying. And then, in order to initiate that APA, typically you have a strong like bilateral TA response, and you do turn off the soleus. Our analysis didn't like bring that to light. I don't know if maybe it was just too like the way we did the analysis or something, it didn't like highlight that. And also in people with Parkinson's, they have fractionated EMG, and they have like co-contraction. So they might not be turning off their soleus sufficiently, but they're also not activating their TA. And you can see that in some of the plots where they have like small bursts and then like quiet periods and then a burst and then a quiet period. And you can see that across the board in Parkinson's, arm muscles, leg muscles, et cetera, all of those are impaired. So like that muscular activity is impaired in Parkinson's. So yeah, I think like the whole muscle coordination is a problem. Yeah. Right. That is a really good question about the spinal cord implication. I have not seen the spinal cord stimulation for freezing. I have heard of PPN stimulation. And I think that kind of went out of fashion. Like I think it looked promising, but I don't think it really was in the end. Certainly like the central pattern generator is very spinally driven. We think that there's maybe like a reticulospinal component, certainly for gait and posture in general in Parkinson's, and also maybe for freezing. But yeah, we don't know if that, like if the reticulospinal system is impaired. I think it might be across the board for Parkinson's or anybody who has like the Parkinsonian gait impairment. But yeah, Column's actually working on that a little bit more. So hopefully we'll have more soon. Right. That is a great question. I don't know that anybody has looked at breathing during freezing. I know that they've looked at heart rate variability, and that is increasing immediately prior to a freeze. I'm not really sure how they do that super reliably because the heart is so I don't know how long they like record that. I don't know if that's related at all, but I don't know of anyone who's looked at breathing. So it'd be really interesting. Mm. Right. Yeah. I think that's a great idea. As a dancer, I was trained to couple my breathing to my movement, but it's never really occurred to me to think about that in terms of gait. So thanks. I probably will. I'll report back. Yeah. Yeah.
Was that strictly
I think so. Yeah. Some people, we had it like turned up all the way and they couldn't feel it. And so I think that when it wasn't effective, maybe they weren't feeling it very strongly, but we didn't really have a way of comparing like the intensities across cueing modalities. I think that like vibrotactile, maybe even on a different limb would probably be sufficient. But just down by the ankle, it was just probably not sensitive.
when you first presented? I have almost to this question with all, but that would introduce a lots of lifting. That would be fun.
Right. Yeah, I think that's why they put it on the ankle to try to have that like leg response, but more as a cue and not as like a reflex. So I think that was kind of part of the rationale. But yeah, it just didn't work. But I don't know, like if we did a reflex, would it work? We had at one of those slides with the self-cueing slides, we had a postdoc do like part of his dissertation with us, and then he came back for a postdoc. And he had like a ankle orthotic, and it was pneumatically driven, and it went like boop, boop, and tried to like facilitate the APA. Which is not what you're asking, because you're asking like a different response. But like, even if you just like facilitate that APA, it can kind of help, but it was kind of weird for people. So I don't know. Yeah. Great question. I don't know. So Parkinson's is like a loss of automatic movement. So like movement is preserved, but you can't get at it. So the movement planning and preparation is all there, but accessing it is not there. And so like if you ask somebody to like, walk across the room, they might be wheelchair bound. They can't do it. But if a fire alarm goes off, out of the building they go. Like they might just stand up and walk. And so like the fire alarm is like a very intense, externally driven cue, right? And so it's basically like it's a paradoxical kinesia is what it's called. And so cueing is just like a little baby paradoxical kinesia, where you're accessing that movement through like a different pathway than your standard pathway. And it allows you to move better. You can also release APAs using a startle response. So you have a really loud bang and like people will move really quickly. But it doesn't have to be loud and intense. It just has to be loud enough, I guess. Yeah.
So the thought is bypass.
Well, I don't think you can say that you're bypassing the basal ganglia entirely, but certainly you're bypassing the self-initiation portions of it. Yeah. And you're just accessing that movement in a different way. So for people with freezing, if you talk to them, they will say like, oh, I've used this cue or this technique to get unfrozen. And then one day it just stopped working. And so I think like even a cue can become sort of like automatic. And then it goes into the automatic bucket and not the like externally driven bucket. And so they have to try a new type of cue. Yeah. Yeah.
Great talk, but in the video you showed where there's a turning place,
Yes.
I was thinking, what's happening to dopamine driven person that's new
I don't know. Yeah. So I showed that video because in my opinion, turning is a transition, going from one direction to another. Dopamine is a tricky beast in freezing of gait. For some people, it helps. For some people, it does nothing. I think a very small minority, it worsens freezing. I don't know that like dopamine fluctuations are that quick that like that it's not, I'm walking, my dopamine drops, now I'm not walking. It's more like as I go through the day, I may have more or less freezing as my medication slowly wears off. But I don't think it's that instantaneous, but I'm not sure. Yeah. Yeah, so you're talking about, so they have like a Parkinsonian APA, let's call it, whatever that looks like. And then they have a failed toe off. Okay, and so I think like I've made the case that like that failed toe off is a really important part of the transition. And then your question is like, what's happening during the festination? Yeah, and I don't know. I don't think we know at all. But if you look at some of that, like sometimes freezes are very short. So they're just like a single attempt and then a successful attempt. But a lot of times it does trigger like this oscillating behavior where they're just like, they get like into some sort of weird feedback loop until they can somehow break out of it. And I think your question was like, what's causing that? And sure. Yeah, there absolutely could be multiple processes for different types of freezing. So is that festination caused by a different thing than an akinetic freeze where like you have an attempt, but there's just like no movement? Yeah, the answer is we have no idea. and maybe, but it's hard enough to like elicit a freeze in the lab and measure something, heart rate or neural activity. And then in order, like we don't get enough data usually to split by freezing type, although someday I would love to. So we'll see. It's more likely that you would get some differences between like a freezing and an intentional stop or something like that. Yeah, in the literature at least. Yeah. Thank you. Thanks everyone.